Hello everyone, this is my presentation on CRISPR. My name is Mikhail Couvet. Let's get into it. So what is CRISPR? Okay, so before we talk about what is CRISPR, we'll talk about how bacteriophages invade bacteria. So we all know that viruses need other living organisms to reproduce. And they do this by injecting their DNA, and then they use the cellular machinery to create more of themselves. Well, what happens in the case of CRISPR is a bacteriophage comes in, it injects its DNA into the bacteria, and the bacteria will chop up the viral DNA into a bunch of little pieces, and it will keep a piece of that viral DNA and put it into a part of the bacterial genome called CRISPR, or clustered regularly interspaced short palindromic repeats. And the reason why it's called regularly interspaced short palindromic repeats is because every so often in this area of the genome, you have a piece of viral DNA. So you have a piece of viral DNA, a bunch of nucleotides, another piece of viral DNA, a bunch of nucleotides, and those bunches of nucleotides in between the pieces of viral DNA are the same. So that's why it's called palindromic repeats. CRISPR as a gene editing technology was discovered by Jennifer Doudna and Emmanuel Charpentier. They were the first to really show that it could be used as a gene editing technology, and they won the Nobel Peace Prize for this in the year 2020. And so, as we just talked about, as bacteria use it, it's used as a defense mechanism. So what happens is once the bacteria has the viral DNA encoded in its own genome, it can replicate this into complementary RNA, right, through transcription, and it can take this complementary RNA and put it into a Cas protein, and this Cas protein uses the complementary RNA to guide itself over to, let's say, the same bacteriophage is invading the bacteria again, so it also injects its DNA. Well, now this Cas protein has a complement of that injected DNA, so the Cas protein is guided over to the newly injected DNA, and it can disintegrate that DNA and eventually, protect the bacteria from the virus. And so that's the purpose of it in bacteria. So as used by humans, well, we've decided to take this Cas protein and like this system of guide RNAs, and what we can do is we can make our own guide RNAs and put them into Cas proteins, and we can guide these Cas proteins to specific points in the human genome, and by cutting on both sides of a gene, remove a gene, we can of course, if we remove genes, we can put in new genes, and we'll talk about this further in the next slide. So again, we can remove genes. Obviously, that's disabling genes. If we say maybe a protein's being overexpressed, or there's a certain receptor we don't want to be expressed at all, we can go ahead and remove the gene that codes for that protein or receptor, etc., by cutting on either sides of that gene using the Cas protein and the guide RNA. We can also edit genes if we use, typically with Cas9, it cuts with something called blunt ends. So blunt ends are very hard to stick new nucleotides onto because they cut bluntly, right? There's nowhere to actually stick new nucleotides onto. But if we use a Cas protein called CPF1, it cuts with sticky ends, and that makes it a lot easier to go ahead and take a new genetic sequence, like maybe a short set of nucleotides, and stick it on there, because the beginning and end are complementary to those sticky ends. So then we can insert a new gene, and then that allows us to, maybe someone's missing a protein, like they don't have the gene sequence needed to create a certain protein. Well, we can introduce genes that allow them to create that protein. Of course, the bigger picture, right? We always want to talk about the bigger picture when we're talking about new technologies being brought up in medicine. There's been, of course, this is one of a lot of controversy, but there's been talk of designer babies. So let's say you want your baby to have blue eyes, or be a certain height, or be immune to this disease or virus or whatnot. You can go ahead and very early on in the fertilization process, go ahead and edit the genome of a zygote, right? And then you can have all those attributes for your baby. The next thing, obviously, would be fixing genetic disease in living people. So let's say a person doesn't produce enough of a protein, and for that reason they have a genetic disease. Well, maybe we can go introduce Cas into certain tissue, and introduce the genes required to make that protein. or perhaps they have a protein that causes disease in the first place. We can go ahead and cut out that DNA. This really hasn't been used as an application yet. I believe I read a paper like not too long ago that there was the first actual medicinal use of Cas. I can't remember exactly what it was for, but I believe it just started to be used at a very like simple level. And then of course we also talked about preventing diseases. So the last thing I want to talk about is a research paper involving CRISPR. So again, CRISPR is just a section in the bacterial genome. What's really important is the things we've gained from CRISPR, such as guide RNAs and the Cas protein, right? So in this paper, what they did is they predicted the on and off target activity of Cas13d, So the idea here is that you want to use a Cas13d. A Cas13d targets RNA, not DNA. So it targets RNA. And the idea was that you can introduce a certain guide RNA, and then because it's targeting RNAs, it's not like an on-off switch. Like you're not cutting out a gene and turning off that gene, or introducing a gene and turning on that gene. By targeting the RNA, you can actually like treat it more like a dimmer switch. So let's say a protein's being produced at too high of a level, we can just turn it down to 50% because we can attack maybe 50% of the RNA that's being transcribed, and we can turn it down. We don't have to just completely take out that gene that codes for that protein. So yeah, and the idea behind this paper is that if we could train an AI to predict how effective a certain guide RNA will be at guiding Cas13 to the RNA targets that it wants to degrade, we can predict how much will turn that dimmer switch up or down, and which obviously is very important because you don't want to put in a guide RNA. It's going to be 100% effective and completely disintegrate all the RNA, because then you're not going to have any protein, right? We only want to turn down 50%, for example. And so yeah, that was the paper I found that using CRISPR. So thank you so much, and that's my presentation.